Every face in New York, even yours right now, hosts an invisible colony of 8-legged creatures that have evolved beside us for 40 million years. Nearly every adult is a vessel for Demodex mites. They live, breed, and die on our skin without us ever noticing. If this is so universal, why has almost no one ever talked about it? The answer lies in the biology unfolding beneath our noses, and it is far stranger than you think. Under the lens of a high-powered microscope, a single human eyelash becomes a vast landscape. Crawling along its base, a translucent creature, barely a third of a millimeter long, moves with slow, deliberate strokes of 8 stubby legs. This is Demodex folliculorum, one of 2 species of face mite found on nearly every adult in New York City. Its body is elongated, almost worm-like, with tiny claws at the ends of each limb for gripping the walls of a follicle. The second species, Demodex brevis, is shorter and stouter, built for burrowing deeper into the oily labyrinth of sebaceous glands. Both species are perfectly adapted for life on human skin, but each claims a different territory. Demodex folliculorum prefers the open shafts of hair follicles, especially along the forehead, cheeks, and nose. Here it glides through sebum, feeding on skin cells and oils while clinging to the follicle wall. Demodex brevis, meanwhile, disappears from sight, tunneling into the ducts of sebaceous glands, where it feeds directly on glandular cells. This division of labor means that every pore, every oil gland on the face, is a potential home for one mite or the other. Their anatomy is a study in specialization. Both species are nearly transparent, their bodies lit from within by the faint glow of absorbed skin oils. 8 legs extend from the front half of the body, each jointed for gripping and crawling through the narrowest spaces. Without wings or eyes, they navigate entirely by touch and chemical cues, moving at a pace measured in millimeters per hour. The rear segment of their body tapers off, packed with undigested waste, since these mites have no anus, an oddity that will matter later in their story. Dr. Meera Patel, a dermatologist at a Midtown clinic, examines a slide under her microscope. She points out the subtle differences: folliculorum's longer, slender frame suited for the open follicle, and brevis's compact build for squeezing into glands. Under magnification, the mite's limbs flex and grip, revealing a choreography honed by millions of years of cohabitation with humans. Each adaptation, from body shape to limb placement, reflects a niche so specific that neither species can survive anywhere but on human skin. These creatures are not invaders, they are permanent residents, shaped by and shaping the micro-landscape of every face in the city. Their presence is a fact of biology, hidden in plain sight, as ordinary as the pores they call home. A face mite's life begins in darkness, deep inside a single pore. An adult female lays her eggs at the base of a follicle, tiny oval capsules nestled among skin cells and oil. Over the next 3 days, the eggs hatch into 6-legged larvae, each smaller than a speck of dust. These larvae feed and grow, molting once to become 8-legged nymphs. After a week, they reach adulthood, fully equipped for the only world they will ever know, the landscape of human skin. Every stage unfolds within the confines of a pore or gland. Mites never leave their host. They cannot survive elsewhere, not on a pillow, not on a towel, not even for a full day in open air. Their entire existence, from egg to adult, is compressed into about 14 days. The rhythm of their lives is set by the cycle of human sleep and wakefulness. When night falls and the city quiets, face mites stir. Guided by chemical signals and the pull of melatonin, adults crawl out from their follicles. They move slowly, propelled by their 8 legs, covering just a few millimeters in an hour. On the surface of the skin, they search for mates. Mating takes place in the open, often at the rim of a pore or along the oily slopes of the nose and cheeks. After mating, females retreat back into the safety of a follicle to lay their eggs, beginning the cycle again. One peculiarity sets these creatures apart from almost every other animal: they have no anus. All the waste they produce, mostly undigested oils and bits of skin, accumulates inside their long, translucent bodies. Over the course of their short lives, this internal waste builds up, visible as a dark mass toward the rear. When a mite dies, its body breaks down inside the follicle, releasing its contents into the microenvironment of the skin. This strategy, as strange as it sounds, has allowed face mites to thrive for untold generations. Their survival depends on remaining hidden, moving only under cover of darkness, and living their entire lives in close quarters with millions of other mites, each one a silent passenger on the crowded faces of New York. In dermatology, numbers tell a story that the eye alone cannot see. Dr. Meera Patel, a specialist in skin disorders at a Midtown clinic, spends her mornings counting what most people never realize is there. She explains that while nearly every adult carries face mites, it is their density, not just their presence, that matters for health. Under her microscope, a single square centimeter of skin becomes a census tract. If the tally climbs above 5 mites per square centimeter, that is the diagnostic threshold. This number is not arbitrary. Decades of research have shown that when mite populations cross this line, the risk of inflammation rises sharply. Conditions like rosacea and blepharitis often follow. Rosacea, marked by persistent redness and bumps across the cheeks and nose, has long puzzled both patients and doctors. Recent studies reveal that people with rosacea often harbor higher densities of Demodex mites, sometimes 10 or 20 times the baseline seen in healthy skin. The mites themselves are not the direct cause, but their presence in large numbers can trigger an immune response. The body reacts to the buildup of mite waste, fragments, and bacteria, setting off cycles of inflammation that become visible as redness, swelling, and irritation. Blepharitis, a chronic inflammation of the eyelids, tells a similar story. Dr. Patel points to patient slides where clusters of Demodex folliculorum are packed around the base of eyelashes. The result is itching, flaking, and sometimes even the loss of lashes. Here again, the critical marker is density. Fewer than 5 mites per follicle rarely cause symptoms, but higher counts measured during clinical exams correlate with more severe discomfort. These diagnostic rules guide treatment. When Dr. Patel identifies a patient with stubborn rosacea or unexplained eyelid irritation, she samples the skin with a drop of mineral oil and a glass slide. The sample is examined under magnification, and the mite count begins. If the numbers cross the threshold, she knows the skin's microecosystem is out of balance. Treatment may include topical medications or special cleansers aimed at reducing the mite population back to its invisible baseline. Demographics matter too. Mite density peaks between ages 20 and 30, when sebum production is at its highest. Older adults, especially those with larger pores, also show higher counts. Immunocompromised patients, those with weakened defenses, are at particular risk for overgrowth, sometimes experiencing severe inflammation that resists standard therapies. But for most, the mites remain unnoticed, their populations kept in check by the skin's natural barriers and immune patrols. Dr. Patel emphasizes that the presence of mites is not a sign of poor hygiene or disease. Only when their numbers surge past the clinical threshold does the balance tip toward pathology. For the vast majority, these creatures remain silent, invisible, and harmless, an ordinary part of the skin's hidden world. Rush hour in New York creates a living network of contact. On a crowded subway, faces pass within inches. In the city's salons and beauty bars, brushes and tweezers move from client to client, sometimes in quick succession. Each of these moments is a potential handoff, not only for bacteria or viruses, but for the invisible passengers that have evolved to travel only on human skin. Salon culture thrives in New York. Eyebrow threading, lash extensions, and makeup applications are daily rituals for thousands. Shared tools, even when wiped between uses, can retain tiny traces of sebum and skin cells. While Demodex mites cannot survive long off a living host, the brief window between clients is sometimes enough. Studies have detected mite DNA on communal beauty tools, though live transfer is harder to prove. The risk is not dramatic. These mites are already present on nearly every face, but the city's beauty routines create extra opportunities for mixing populations, especially in venues where speed and volume take priority over deep sterilization. Dr. Jonah Kim, a molecular parasitologist, has spent years examining the genetic fingerprints of face mites collected from New Yorkers. His lab sequences mitochondrial DNA from mites plucked off volunteers of all backgrounds. The results reveal a hidden map of ancestry. Demodex clades, genetic lineages labeled A, B, C, and D, cluster along lines that mirror human migration. A volunteer with West African heritage carries clade C, a lifelong Manhattanite of Italian descent hosts clade D, while a recent arrival from Southeast Asia harbors clade B. These lineages persist even after generations in the same city, suggesting vertical transmission within families is still the main route. Mites pass from mother to child, often in infancy, and then remain for life, rarely mixing with new arrivals except through close, repeated contact. Yet New York's density and diversity create rare moments of cross-traffic. Dr. Kim's team has identified a handful of cases where a person's mites do not match their known ancestry, an Asian American roommate carrying a European clade, or a Bronx teenager with both African and Latin American lineages. These outliers are rare, but they hint at the city's unique role as a biological crossroads. Dating, shared apartments, and beauty treatments all blur the boundaries between ancestral lines. The subway, with its sea of faces pressed together day after day, may be the largest living experiment in accidental mite exchange ever assembled. Despite these findings, there is no city-wide system for tracking mite populations. The Department of Health monitors outbreaks of lice or scabies, but face mites, universal and symptomless in most, fall beneath the radar. No regulations require salons to test for Demodex, and few dermatologists discuss them unless pressed by a patient's symptoms. The absence of official surveillance leaves a gap in understanding. In a city where nearly every adult is a host, the true scale and movement of these ancient lineages remain largely uncharted. The result is a blind spot, one that raises questions about what else goes unnoticed in the daily churn of urban life. A single face can produce enough sebum in a day to fill dozens of mite bellies. Demodex folliculorum and Demodex brevis, long cast as freeloaders or even pests, may play a quiet role in keeping the skin's delicate ecosystem in balance. As they move through the labyrinth of pores and oil glands, these mites consume the excess sebum that would otherwise build up on the surface. Their appetite for oil is relentless. Each mite feeds on skin cells and lipids, gradually thinning the film that coats every face by nightfall. In a city where pollution, stress, and cosmetics can tip the skin's balance, this steady consumption of oil may be more helpful than harmful. But their diet does more than just tidy up. Demodex mites also ingest bacteria that thrive in the oily environment of the skin. By grazing on these microscopic neighbors, the mites may help keep populations of potentially harmful bacteria in check. Laboratory studies suggest that their presence can reduce the growth of certain pathogens, including strains of Staphylococcus and Streptococcus, which are known to cause skin infections. Their digestive enzymes break down complex oils into smaller fatty acids, byproducts that, in turn, create a less hospitable environment for invaders. This hidden maintenance crew works in silence, invisible to the human eye. The average adult face carries hundreds to thousands of mites at any moment, each one performing its task without notice. When populations remain low, their movements and feeding leave no trace, no redness, no swelling, no sensation at all. Rather than invaders, they act as caretakers, consuming what the skin sheds and helping to regulate the oily microclimate that would otherwise favor less benign organisms. For most New Yorkers, the vast majority of these mites will never cause harm. Their numbers rise and fall with the rhythms of sebum production, quietly adapting to the needs of their host. In this way, Demodex mites may be as much a part of healthy skin as the bacteria, sweat, and dead cells that make up the city's invisible, ever-changing surface. New York's 8 million faces form a living experiment in biological mixing, shaped by the city's relentless churn of people and cultures. In the city's densest neighborhoods, population density can exceed 70,000 people per square mile, far higher than most places on Earth. Every packed subway car, every crowded apartment, every fleeting brush of skin in a coffee shop or on a dance floor becomes a point of contact in a vast network of human proximity. But the story of face mites in New York does not begin with adulthood. It starts in infancy. Studies show that newborns arrive in the world without these microscopic passengers. Colonization happens in the first weeks and months of life, usually from close contact with parents or caregivers. A mother's cheek pressed to her child's face, a father's kiss, a shared pillow. Each moment offers a bridge for mites to cross. Once established, these lineages often remain remarkably stable, persisting for decades. Most adults carry mite clades that mirror their ancestry, passed down quietly through generations. Yet the city's social rhythms complicate this inheritance. Shared living spaces, frequent roommate turnover, and the rapid pace of dating in New York all create new opportunities for mites to move beyond family lines. In a typical year, tens of thousands of New Yorkers change apartments. Dating apps drive a constant flow of new connections, compressing what might have been years of close contact into a handful of weeks. Each new relationship, a shared bed, a borrowed towel, opens a window for horizontal transmission, allowing mite populations to remix in ways rarely seen in more stable or isolated communities. Despite this, the true extent of urban remixing remains an open question. Genetic studies reveal that most people still host mites matching their own ancestry, even after generations in a melting pot city. Only a handful of cases show evidence of clade mixing. A Bronx teenager with both African and Latin American lineages, a roommate pair with mismatched mite ancestry. These exceptions hint at the city's unique ecology, but no one knows how often such exchanges occur or what they mean for the biology of the mites themselves. Urban density, social churn, and the intimacy of daily life in New York may be quietly reshaping the genetic landscape of face mites. But without systematic tracking or citywide surveys, the scale and significance of this remixing remain hidden. In a city built on connection, the invisible world of face mites poses questions science has yet to answer. Every face in this city carries a private ecosystem, one that science confirms is nearly universal, yet almost never discussed. As urban density grows, our invisible companions adapt alongside us, reminding us that human identity is always shared, never truly solitary. The microscopic world doesn't wait for our awareness. It's part of who we are right now. What else are we still carrying, unnoticed? Thanks for watching.